Hello everyone, today we are going to start the hallmarks of the cancer. The cancer is caused by the multi-sequent DNA mutation that affect the fundamental cellular processes like growth, DNA repair, and survival. Due to these features, we can differentiate the cancer cell with the normal cells. And they show some hallmarks and features, and we are going to talk about them one by one. The first is a growth signal self-sufficiency. It is a hallmark of a cancer refers to the ability of cancer cells to proliferate independently of external growth signals. The normal cells require signals from outside to grow and divide, but the cancer cells then become autonomous, generating their own signals or ignoring the external cues when inhibitors. And this will happen due to mutations in genes encoding. It can be due to proto-oncogenes that is responsible for the cell proliferation that increases cell growth. And due to overexpression of proto-oncogenes, there will be increase in growth of dead cells. Taking the growth factor receptors. Growth factor receptors also constitute for the signal most likely HER2 in the breast cancers. These are signaling molecules. These some signaling molecules like the ROS proteins. These are the small guanosine triphosphates. You can also say GTP-6 that act as molecular switches regulating cell growth, differentiation, and apoptosis. And due to that, these are colorectal cancer, lung cancer, and pancreatic cancer. And transcription factors like MIC, having two types like MIC-C and MIC-N. Okay, there are two types, MSC-V and N. The MSC-C causes the Burkitt lymphoma, and MSC-N causes a neuroblastoma. Okay, and this is said cyclogluteus. Cyclogluteus also responsible for cell proliferation in diva cyclins and cyclin-dependent kinases. Cyclin-dependent kinases and the other key regulator of the cell cycle, ensuring its progression through various phases and maintaining genomic stability. Actually, these cyclins activate CDKs, which then phosphorylate and target the proteins and driving a cell forwardness cycle. And these CDKs are the family of kinases that are inactive until they attach with cyclin. The cyclin-dependent kinases are inactive, but it will become active when they are attached to cyclin. The second hallmark is anti-growth signal insensitivity. The anti-growth signal insensitivity mutation in tumor suppressor gene. Anti-growth signal insensitivity is basically mutation in tumor suppressor cells, tumor suppressor genes. What is happening if you guys are more than G1 phase and cell cycle and the S phase? In both phases, it's a replication phase for the DNA. It is in this phase, it's a replication of DNA. Between this point is a checkpoint that is controlled by RB proteins. These proteins are responsible for the checking the cells are okay and they are proper and they are having no defect, there is no abnormality cells to divide and it is not cancerous cell. The checkpoint is controlled by RB proteins. If there is a defect or inactivation of RB proteins, then there will be mutations and there will be cancer cell growth. Because this is a checkpoint, so many of cells are abnormal, they're not able to grow, but we are sending them to atrophy that will become dangerous, and in the future they will become cancer cells. And we'll see how these will be active. RB is active, but after phosphorylation of RB become inactive. The phosphorylation of RB causes the inactivation of RB proteins. The next thing for the anti-growth signal insensitivity first, tumor suppressor gene. If tumor suppressor genes are inactive, then there's no Other thing that can stop the growth of cancer, taking loss of E-cadherin function. The E-cadherin is a tensional and calcium-dependent transmembrane glycoproteins that play a crucial role in cell adhesions, particularly in epithelial cells. And it also acts as a tumor suppressor and by inhibiting the VNT signaling pathway. Inhibiting the VNT signaling pathway, and also to be responsible for the cell contact of cells. Like two cells are attached to each other, and it's mostly responsible thing is E-cadherins. Okay? And when the E-cadherins lose their function and leaves lots of contact inhibition due to enough to mutation, there are many of cells getting the separating. So if one cell is in a group, one cancer is in a group with cells, then E-cadherin lost their function, then these cells will separate to different places. Our next hallmark is evasion of apoptosis. Inhibition of apoptosis. In the apoptosis, we already know there is some pathways, extrinsic and intrinsic pathways. Okay, you guys know there's a BCL2 expression or a liking. If BCL2 expression is increasing, there's no release of cytochrome C. Cytochrome C is not releasing, there will be no apoptosis. Same happening in mutation their genes, they regulate the apoptosis and due to regulate the BCL2 and TP50G defect, and it will result in follicular B-cell lymphoma. You defeat the cell will not go to dead process and it will be start growing, and it will result in the cancer. Then they limit the replicative potential. Three, these cells are reduction of telomeres. Telomeres. Actually, telomeres are responsible for the maintenance and lengthening of telomeres is beneficial for the cell, for the dividing. Like, as longer the telomeres is, the cell will divide as much. Okay? The shortening of telomeres in unable to divide, the shortening of telomere did not able the cell to divide more. So it is just like a aging. Cell aging, the cell aging can be detected by the telomeres, but in the cancer, there's a reactivation of telomere to divide. There's a prevention of chromosome shortening and cell aging. So always the cell can still become young and it will divide and divide as much it can. So the females are using botulin and Botox for the anti-aging process, but the cancer cells are using telomerase reactivation to prevention of chromosome shortening and cell aging. Understand? Next is sustained angiogenesis. Like cancer cells also want some nutrition, oxygen for survival through they make new blood vessels. They survive for their survival they make new blood vessels, and that process is called angiogenesis, and for that the responsible factors are VGF and FGF, the fibroblast factor. Both factors are important factors for the angiogenesis and they are responsible to divide new blood vessels formed, and cancer cell will take their happy meals like nutrition, oxygen, and the thing they want. Okay? And these vessels can be separated from existing capillaries like new angiogenesis or endothelial cells are recreated from the bone marrow and with the vasculogenesis. The vessel may be leaky or dilated. It depends on the feature of the cancers. But the angiogenesis is a more important point, what an important factor that VEGF and FGF. And our third factor is the Weinberg effect. The Weinberg effect is that taking of glucose with the cancer cell. How much glucose is cancer cell taking? Okay? And this is a shift to glucose metabolism away from mitochondrial oxidative phosphorylation toward the glycolysis. It is a property for cancer cell is take high quantity of glucose from the metabolism. Okay? And for generating high number of ATP. For that, and we are using this process. We are using this thing in PET scan. In PET scan, but what we are doing, we are giving radio-labeled glucose. When you give the radio-labeled glucose, the abnormal cell will take high amount of glucose of that. And that will be shown on PET scan by a shiny colored slit, okay? Again, it's a very important point for the PET scan, which effect is using the Weinberg effect. The Weinberg effect is taking of glucose and how much glucose the cancer taking, and by PET scan we can detect the cancers by giving that radio-labeled glucose. And they are saying aerobic glycolytes provide rapidly degrading cancer with the carbon 80% tissue cell structure, and it leading to increasing lactic acids. Taking immune evaluating cancers, actually immune cells also detect cancers and destroy them. Okay? It is one fact that cancers are independently, but the immune cells having the capability to destroy the cancer cells, but they have to do something for their survival. At this we are also doing for our survival many things we are doing. Like in winters we are taking the jackets. In summers we are wearing some cotton clothes like that. Seem like that the cancer learns making them adaptations to fight against the immunity and on escaping from their attack. For example, someone found to hit you. You are not a capable person. You are not fine to fight against them. Then you have to escape. Seem like the cancers are doing, and they are doing some escaping system. How they are taking the modification we are read now. Normally, that means they can recognize and attack the tumor cell. For successful tumorigenesis, the tumor cell moves evade the immune system. Multiple escape mechanisms exist. The first one decreases molecular histocompatibility expression. If the tumor cell is now showing MSC class 1, then tumor and then the T cell did not recognize them. You too unable to detect MSC class 1. And due to this expression, lack of this expression, they did not recognize tumor cell and T becomes safe. And cytotoxic T cell unable to recognize the tumor cells. It's a simple thing. If someone is showing MSC class 1, the T cell will detect them and destroy them. But when the cancer cell is not showing decreased expression of MSC, the T cell did not recognize them, unable to recognize, and they will not destroy them. And second thing is, tumors is secrete immunosuppressive factors. That tumor growth factor beta and regulate T cell to regulate the immune response. These are some responses that are anti-inflammatory. Anti-inflammatory response due to which automatically your immune system did not respond well, and they will then chill. Everything is normal in the body. But the third thing, the tumor cell circulate immune checkpoint molecules which inhibit the immune response. This is an important thing. First, you have to know what are immune checkpoint molecules. Immune checkpoint molecules are proteins that regulate the immune system, preventing it from attacking normal cells and tissues. And they act as a brake in your immune system, ensuring that immune responses are balanced and don't cause excessive damage. How do tumor cells extract immune checkpoints? Okay, the tumor cell can take advantage of these immune checkpoints to evade the immune system. By upregulating increased expression of certain immune checkpoint molecules, tumor cell can send inhibitory signal to immune cells, such as T cell, that would otherwise recognize and attack the tumor. The key players for that, for inhibiting the cells, are PD-1. PD-1 this is called a programmed, that one, a protein on T cell, and is a prisoner, T cell. Okay? PD-1 is a programmed. That one. And these are present on T cell and that when bounded ligand PTL1, the T-cell activation and function inhibits. For inhibiting the T-cell, these are one ligand on the T-cell that a PD-1 program that one when it attached with the PDL1. PDL1 is a program that ligand 1. This causes inactivation of T-cell, okay? And the third molecule is CTLA form. It is a cytotoxic T lymphocyte antigen, cytotoxic T lymphocyte antigen, and this is another protein that regulates T-cell activation and can be targeted by tumors to evade the immune responses. Don't forget it's an important thing, it's also present in another topic, like PT1, PDL1. The PT1 is present on T cells. It will attach with PDL1 and become inactive, and there is one more protein, antigen CTLA4. Okay? Now we are doing in next part that the tissue ingredient. The tissue ingredient is due to lose ecadine function road ID. No, the cell contacted due to ecadine. When the ecin helilosis function, given the losing of intracellular junction and the metalloproteinase, degrade the basement membrane and ECM. What's the ECM? The ECM is the extracellular matrices. These proteins are crucial suction signaling component found in a space around cells and in tissues. They provide support, facilitate the therapy and influence the cell behavior through interaction with cell surface receptors. And due to these, with also metalloproteinase enzymes action like collagenase, hydrolysis, this all enzyme will act on basement membrane and it will disrupt it to develop cells attached to ECM proteins like lamin, fibronectin, and cells migrate through degraded ECM and then the revascular dissemination. Okay. And don't forget the ECM protein. The ECM proteins includes collagen, elastin, fibronectin, laminin, proteoglycans, and other also like osteoportin and fibrinogens. These all proteins are ECM proteins, and it is happening in vegan how? First, ecadine will lose its function after there will be metallo. Proteinase enzymes act on basement membrane after the disruption. The cell will start invasion and start dissemination in vascular system. Understand? Now we move to the last hallmark of cell consistent that is metastasis. What is happening in the metastasis? In metastasis, the cell is going to separate to other organs, and tumor cells will embrace separate with a lymphatic supplet. It happens at the end we don't hit him, and that's an extra vein and the harming. The site of metastasis can be predicted by the site of primary tumor, as a target being often the first encounter capillary blood. Some cancers show organ tropism, but the organ tropism means the tendency of the cancer cell to the perfectly separate to specific organ tissue. This means that not all organs are equally likely to harbor the metastatic tumors. Some may be favored over others. Okay? Like if there's a prostate, then it is going to burn white cells, right where veins. And if it is like GIT, it is going to leave our lungs like that. Okay. If any blood spread, it first going to lungs like that. Every organ have their own specific first site of metastasis. And for that things, we have to be known their origin, how they are going to spread. And secondly, there is some other processes, some other important points I want to share in. Like already you guys know there are two types of spreading by lymphatics or by the blood. There's some important point like sarcoma. If you guys know the sarcoma is a within gynecology cancers, and these spread via blood hematogeneously. Okay, like artery thick wall, and you guys know the arteries having the thick walls, and due to which they the difficulty to penetrate. And secondly, the veins, when the thin wall, they can easily penetrate and leave along the most common sites of hematogenous spread. So the sarcoma cancers are going to spread through blood, and if any cancer is spreading from the blood, then it most commonly go to liver or lungs. Understand? And what is our second route? The second route is lymphatics. The carcinoma. The carcinoma is spread through lymphatics. But these are exceptions. Exceptions are for carcinoma spread by bloodstreams. For the choriocarcinoma. Second is renal cell carcinoma. That is spread by renal vein, then hepatocellular. Again, hepatocellular carcinoma, that's a place by portal vein. And one is follicular. Thyroid carcinoma. Okay? Already I done with that quality. In medicine is the important thing. The roots, there are two routes of spreading the cancer for the lymphatics and blood. The sarcoma within gynecological cancers are spread by the blood. And whenever any cancer is spreading by blood, it will go to liver or lungs. And all the carcinomas are going to lymphatics route, but there is exception of all cancers: choriocarcinoma, renal cell carcinoma, hepatocellular carcinoma, and follicular thyroid carcinoma. And it's all about the metastases and hallmarks of cancers.